Hello, everyone. Welcome again to our seminar series on the dialogues on ethology and behavioral ecology seminars. And this series is hosted by the Department of Biological Sciences and specifically by the Unit of Ethology of the University of Pisa. And today we have a very exciting seminar by a colleague of ours from the University of Tuscia, Professor Claudio Carrere, and he will be talking about animal personalities, the power of the individual. I like this title. Anyhow, just a little bit background on Claudio. Claudio got his bachelor/master of science degree in 1995, and then a few years after, he got a PhD in Groningen in the Netherlands. He's somehow, I don't know if it is correct to define him as a non-ethologist or an ethologist that is specialized in birds, we would say. But he worked on common swift and then great tits, Parus major. And then after his PhD, he went to Belgium in Liège. I don't know the pronunciation, but I think it's something like that. And he was studying neuroendocrine mechanism underlying sexual and individual differences in birds. That was very cool. And soon after that, he did a second postdoc, somehow monitoring and coordinating a project, a European project, on understanding patterns of animal group movements. After that, from 2010 to 2015, he has been adjunct professor at the University of Tuscia. And in 2016, he got a Marie Curie in Paris. Finally, in Paris, he was studying the collective personalities in ants and starlings. That is, I didn't even know that there was such a thing as a collective personality. So this is very cool. And only in 2016, he got a permanent position as associate professor at the University of Tuscia. So I really want to thank Claudio for having accepted our invitation. And I really look forward to hearing what he's going to tell us about animal personalities and the power of the individuals. Thanks a lot, Claudio. The podium is yours.
Thank you, Alessandro. Thank you to the audience for listening to me in this on this Wednesday. So, well, thank you for your words. And I'll start focusing on the individual because, I mean, in this period there is a lot of discussions, even among common people, about what is speciesism and anti-speciesism and this kind of things. I don't want to go into these discussions, but certainly I want to put emphasis on the individuality. Emphasis that was given by a journalist of the New York Times several years ago when the study of animal personality was still at its, I would say, infancy. And the cover page of this article, the cover was this nice and provocative picture of modified picture of flamingos, and that are labeled each with an attribute that we would say it is quite anthropomorphic, because there is calm, methodical, curious, introvert, aggressive, shy, and pink. And so going to more recent social media communication, when it appeared on my Facebook, probably this picture, I was a bit annoyed because it was a bit sexist, but second, I said, no, that's not possible that this is for males. This is probably 4 bold individuals and 1 shy individual, or maybe it could be also that 4 are just very hungry, and the other is very satiated. Well, individual differences. What will I speak about in this talk? But first of all, Historia Magistra Vitae, so Historia Magistra Ethologiae, I would say ethologies. But anyway, I would like to go back to some roots of the basic complex, also with examples. Then I will describe personality differences in different species. As Alessandro said, I started as an ornithologist, but actually I have many more species, especially more recently. We will see ants, octopuses, and the third part will deal with eco-evolutionary aspects. So, and the fourth part would somehow outline what are my, and actually the main current and the next research avenues on this topic. Well, I'll just start with a neuroanatomist, Franz Josef Gall, who, and just let's read together, that what he wrote. As this is a translation by neurobiologist Hans-Peter Lipp. A similarity I observed among the tame and wild animals, of which I always kept a few around me, a similar variability of behavior as in humans. A given dog would start hunting almost on his own, while another from the same species and litter remained untrainable. And then in the bottom, thus, after other example, I had to conclude that such properties, so such individualities, were innate in human beings as in animals. With that, however, one was led to ask for the foundations of such innateness. Well, Darwin himself emphasized the importance of the individuality. No one supposed that all individuals of the same species are cast in the same mold. These individual differences are of the highest importance to us, for they are often inherited. Well, the Greek system had divided personality in choleric, sanguine, phlegmatic, melancholic, and Ivan Pavlov, Russian neurophysiologist, based on the studies of his dogs, did the first systematic typology in a non-human species, and he was kind of attributing the final type of personality designation as excitable, lively, quiet, and individual, which resembled somehow the Greek system. And most intriguingly, he hypothesized something that has been tested in these years. I won't speak too much about that, just will mention, is about the relationship between the individual personality and the individual cognitive abilities or cognitive styles. So he was hypothesizing that these kinds of personalities would be marker of different styles of associative learning. I'll leave you with this question. And going forward to more recent times, this is Peter Slater, an ornithologist, actually, an expert of bird song. And bird song, we know, is very much individual, especially in songbirds. There are dialects. And he wrote in this seminal chapter that I really suggest to get, if it's not easy to get it, individual differences in animal behavior. This sentence which impressed me: the average animal emerging after statistical treatment may possess a set of features that are not possessed by any single individual in the group. So this individual has never existed or could never exist. So what are we measuring? Or what are we actually, what are we measuring? What are we representing? And a famous paper by Tony Williams, The Tyranny of the Golden Mean, is a recommended paper to all students of these topics. Well, this is what Peter Slater meant. Now, this we have the behavioral assessment, the tonic immobility test in hands of 2 lines of laying hands. Tonic immobility is a kind of anti-predator response. Anyway, it doesn't matter so much, but you can see how the representation can, representing only the averages, masks important individual differences. And these extreme individuals that are on the right side, this is not noise. They exist. So they may have a role. They are more rare. Well, the first, as far as I know, assessment of, and the first time the term personality appeared in a scientific journal is the Journal Comparative Psychology in the early 90s by Jennifer Mather and Roland Anderson. For the first time, found clear axis dimensions of personality. They call them activity, reactivity, and avoidance, something we have repeated now recently with Jennifer herself, who came to visit us a few years ago, on the octopus. And it's curious this, because the octopus is an invertebrate, and we know it's an invertebrate which is now protected because it's included in the legislation of protection for animal experimentation. Well, after my PhD, I organized a symposium for the European Conference of Behavioral Biology, and surprisingly, the symposium was about animal personalities. We were at the beginning, and this symposium was attended by, I was very shy and surprised that I'm behind in this picture, and this is Felicity Huntingford, this is Pat Bateson, and other names that are now rather familiar to animal personality students. And this resulted in a seminal special issue in 2005. Well, after that, we decided with Dario at the same meeting, we came with the idea that at some point we will have to do a book about that. And collecting most of these people, the book came out in 2013, and it's a kind of compilation of animal personalities studies. By the way, incidentally, Dario Maestripieri is in Rome. I met him last week, and he will is having a short sabbatical, visiting scientist in Rome. And tomorrow, after tomorrow, he will give a lecture at the Department of Philosophy on the evolution of personality. It will be in Italian. And so incidentally, I just put it here, but I will give you, Alessandro, the information. I will send you the announcement. Well, what are we talking about? Well, first of all, nothing new. I or Dario or these people, we have not discovered anything particularly new, but we have just emphasized something which was already known in behavioral ecology. Different sets of literature speak about the same issue, so we can have, terminology is vast. Reproductive strategies, boldness, coping styles, and more from the stress physiologist. Personality, of course, temperament, behavioral syndromes, behavioral profiles, behavioral polymorphism. Well, we are more or less speaking about the same thing, and thing that has to be measured with behavioral tests, often accompanied also by physiological assessment, especially peripheral, that measure individual and consistent, and the word consistent is very important, behavioral strategies, patterns in response to aversive or stressful challenge. Consistent behavior, because when there is a challenge, these differences emerge better. This is rather common. Consistent behavioral traits across lifetime and context, early behavioral tendencies during juvenile age. A practical example, Felicity Huntingford in the book chapter of our book. Now, how do you measure boldness in fish? You can measure the emergence time from a shelter, and then clearly you observe that consistently some individuals are fast emergers, some individuals are slow emergers. Those that are faster mergers are also those that inspect more a novel object, and that are also those, and this is more in the becomes a trade-off for the individual in a situation of risk. They are willing to take more risk. And this is the most exciting part, because individuals have to cope with challenges and risks in everyday life. And there can be different strategies. I mean, I will simplify it. It seems that there are two strategies. Of course, there are a lot of situations of intermediates. It's a gradient, it's a continuum. But sometimes there are bimodal distributions, indeed. We'll see. Well, it's important to remind that I would say a requirement for personality, is a statistical requirement, is that a behavior has to be repeatable. So what is repeatability is that the fraction, the proportion of the behavioral variation that is due to differences between individuals. So when individuals behave consistently across time, and when they behave differently from each other, so the behavior is repeated. So there are individual differences. Now we'll go into more depth into that statistical part. And, well, the issue. Individuals, of course, do not always express the same phenotype. So we have within individual variation. Let's call it individual plasticity. I put this part in bold because this is what I operationally define as personality. Individuals differ consistently between them over time and across contexts in their average levels of behavior, I would say, the physiology, fitness, and this is what we call between individual variation. Of course, individuals differ also in their plasticity. I won't speak too much about plasticity, but clearly this is related. So which is an individual by environment direction, which is potentially resulting in both within and between variation. Well, there is also measurement errors. This I put away this part, but anyway, the total phenotypic variance is the sum of all components. So a true phenotypization has to include all these. Well, let's put graphically this. Well, what do we have here? We have the same data set. Where do we have the personality? So this is a graphical illustration of variation. So above, we have behavior in environment E1 against behavior in environment E2. And in B, we have a correlation. The same data can be plotted with a reaction norm approach. And you see, in B, we have these lines, more or less straight. So in B and in D, we have personality, because there is a correlation and individuals show relatively limited plasticity in D. Well, it is important to say that if between E1 and E2, the behavior change, for example, they all increase, there could be still repeatability and individual differences, because it's the rank order that remain unchanged. Okay? Well, empirical example. Cuttlefish at the University of Tuscia. So we have here same behavior in different tests. We have tests, an alert test, and a threat test. They are similar but different. So they are just different tests. I won't go into the methodological details, but there's high correlation. Different behaviors, same test. This is the same test. Individuals that move during the threat test, they also change color more often during the threat test. Well, let's now, I will always keep the Tinbergenian approach now, from proximate to ultimate causation. But I won't go into too many details on the proximate causation of individualism, but I will give you just here an example. This example comes from my neuroendocrinological studies. And look at this. Well, that needs a bit of explanation, but don't worry. I mean, what we have here, these are males of Japanese quail. The goal of this experiment was to look at sex differences, but this here I plotted only males. What this arrow RLF thought to represent? Well, this is, well, I have to tell you that for the activation of male sexual behavior, it is necessary that in the brain, the testosterone is converted into estradiol. And this occurs thanks to an enzyme, which is called aromatase. This aromatase is present in an area in the nucleus of the hypothalamus, which is called the preoptic nucleus. But this preoptic nucleus, which is the nucleus which is on the image, it projects to the mesencephalon, and in an area near the cerebellum, the periaqueductal gray, where it then projects to the spinal cord and it activates copulation. Well, this plot represents these fibers in the males, and there is variation. So this plot represents the connection between the preoptic area and based on a track tracing study. I don't want to go into those details. And on the y-axis is the intensity of neck grape behavior, which is a copulatory behavior. Now, the more the projections, the more the intense is the sexual behavior. That means that there are males that are somehow more males and males that are somehow less males. Individual differences, deep proximal basis, but there is differences in these projections. Well, why is important to study individuality and consistent individuality, so personality? Well, of course, it is important. I mean, as the Franz Joseph Gall and Darwin himself said, well, what is the nature of these differences? Are there traits that are linked? Are there common underlying mechanisms? Then, of course, the plasticity, the developmental aspects. Does the personality change across life? Is affected by factors during early ontogeny? What are the contributions of the genome environment in shaping the behavioral personality profile, which is assumed to be rather crystallized in adulthood? And then, of course, what is the function and the evolution of personality? How and why is this variation, this huge variation, which was considered as noise before, maintained? Does it pose constraints for evolution? Does it put limits to plasticity? Of course it does. But does also play an active role in evolution. An active role could be as a driving evolutionary force, but behavior we know is a driving force. But it could be also driving evolutionary inertia. So could also buffer, stop evolution. And then in the biomedical, especially literature, it's important. I mean, everybody knows that different people react differently to disease, react differently to stress. Different personalities react differently. This brings to the next issue, which I won't deal with in this talk. But clearly, it is an animal welfare issue because captive conditions animals cope with captive, individuals cope with captive conditions differently. Well, also conservation biology, reintroduction programs. What would you reintroduce bold animals or shy animals, or it has to be tailored, it has to be taken into account. Well, this is just a schematization of on how if an individual could have a cognitive and emotional, different cognitive emotional responses, physiological responses, and even in different health outcomes. But I bring you immediately to a seminal paper, a seminal study of Sonia Cavigelli and Martha McClintock. And this is a study that I always give to my students because it's quite paradigmatic and it opened important avenues of research. And Sonia characterized phenotypically two groups of rats, neophobic and neophilic. And she followed them longitudinally for the whole life. And every now and then she tested them and she measured the hallmark of the stress response, so of the HPA reactivity, the corticosterone. And this corticosterone was a level where higher consistently in the neophobic individuals. And eventually the shy, so the neophobic are the shy individuals, did die sooner. Well, that one does not mean that who is shy doesn't want to make predictions or so, but this was very interesting because then other studies explored the causes of the death. They were more subjected to tumors and other things, so it opened it. But just to show you this longitudinal studies. And this brings me to the issue that, of course, methodologically, studies of personality is very demanding because the sample size should be high and the animals have to be studied longitudinally. So now our repeatability. Of course, it's easy to measure repeatability after 1 week, but we would like to know it repeatability after 1 year or more, or even for the whole life. Well, let's go into this characterization part. And let's start with my species of the PhD, the great tit. Great tit is a small songbird, very common, whose behavioral ecology is well known. And it all started with Monica Verbeek in the 90s, discovered consistent individual differences in two what we called once behavioral domains, in exploration, so in a non-social task, and in a social task, in aggressive behavior. Great tits are highly territorial birds. You know? And from this characterization, a first characterization, started the idea and the project, the Dutch project, to build selection lines for these traits, exploration and aggressive behavior, actually exploration. So what are the tests of exploration, which were then the criteria, the selection criteria for forming these selection lines on which then I worked for my PhD. Clearly, when you have selection lines, which are first the tests? The tests were very fun. You know, the two tests of novel object, approach to a pink panther, so latency, so it lasted 2 minutes, of course. Consider that if you do the same with corvids, it can take up to 2 days. So you have to put a cutoff of your test, not at 2 minutes, but at least at 1 or 2 days, because these are very neophobic animals. So you have to adjust the tests just to make an example. Another test, the pain light vector approach to a novel object. And well, what Monica found was that clearly there were many individuals that were very fast, relatively few individuals that are very intermediate, and many that are very, very slow. Of course, this could be not really bimodal distribution, it could be more, and this is a limit of all these studies. It could be a Poisson distribution because we don't know when they would react. Maybe they would react. So the group on the right side is certainly more heterogeneous. Another test, which yielded similar results, was the speed of exploration of a Roman room. The animals were allowed spontaneously to enter in this room, and they were allowed to explore the trees. And those individuals that were fast explorers, they jumped from tree to tree, and they covered all the tree within a few minutes, in a short time. While the slow individuals, the shy, let's say, they were jumping, they often did not complete to cover all the trees, but instead they stayed on one tree and they jumped on the small perches of each of the three. Somehow, this led to the hypothesis that the slow animals are more thorough explorers, and the fast animals are more superficial explorers, but they are faster. They may even take two risks. Well, many other studies came, so these things were somehow in part replicated in other species. Anyway, on this test, the selection line started, and the selection lines rapidly diverged with the realized reusability, so based on generations, changes on different generations. Was quite high, 0.54. And I worked basically characterizing, because in what other things these individuals that look apparently all the same, so these are subtle differences, but in what other things they differ. So the exploration score on the y-axis is a combined cumulative score of the all these tests that that I have illustrated. Now, I summarize here part of the characterizations. Clearly, these animals that differed, of course, in exploration also differed in aggression. So the aggression exploration syndrome, and then this is a syndrome, when two domains, they correlate. So you are fast explorer, you are also an aggressive individual. In fact, the fast explorers have a short latency to attack an intruder than the slow animals, and the attack rate is also higher, but the threat displays are very low. And so that doesn't mean that the slow are not aggressive. The slow are aggressive, but they deal with the social challenge differently. They did many more threat displays, and somehow they are a bit more diplomatic. So this is a typical threat display. You can see it. Well, very interesting. Now let's see another differences of this line. So it's not spectacular, but here, but it was confirmed by other studies. It's basically a higher reactivity of the HPA axis of the cortical. So basically, the slow the shy are more stressed in terms of corticosteroid response, and this is similar to the pattern of the rats of Sonia Gavigelli, if you recall. Just the dots color are different, but doesn't matter. Well, this led many hypotheses it could be done, but I mean, the corticosterone has an effect also on dispersal. So, and we hypothesized that social stress could lead to differential corticosterone release in the shy and the bold individuals. Of course, we know from the physiology that corticosterone induces gluconeogenesis and activity, foraging, and so this could be a mechanism underlying different patterns of dispersal, of different spatial pattern of the population. Well, there is a long list of parameters in which the two selection lines differ. Some I have are those that I have illustrated. That this is just a list. I won't go into all these details, but I want now to look at consistency across time. If you see the score exploration, so what I had the opportunity during my PhD to test the birds that were kept in standard condition after 3 years. Well, 3 years is a lot in juvenile age and in adulthood, but 3 years is a lot for a great deal. So, and you can see that the slow individuals increased, the fast remained similar, and but so the pattern is intriguing, but also the agonistic behavior was consistent across in this kind above its 3 years interval. Here is a 7 month interval, and this was highly consistent. The same line differed yearly in exploration. And what can you measure when for a bird when you are in the nest? You can measure begging behavior. Begging behavior is the offspring solicitation behavior. And begging behavior, so the one that I reported in the very first provocative slide from the social media of the 4 swallows, one of them was not begging. Well, I measured the intensity of begging, and there was a clear difference between the fast and the slow chicks. So the fast were more competitive, apparently. So this was an evidence of an early line difference, and even something that could indicate the genetic basis for begging strategies. But this is another question. But to summarize, I give you the density of information might be high, so every now and then I try to summarize. Well, the line differences in these personalities encompass many traits. They are present early in ontogeny. They are consistent, but not completely stable over time. And that the slow line could be less stable. And one reason would be this fact that this is a point from the selection process. It may be a bit of an artifact. I self-criticize my data, but I mean, it's not an artifact. It's something that we don't see because of the limitation of the tests. Anyway, it is a more heterogeneous group, but it might be genuine. So it might be that the slow are most flexible. Well, let's go to the ontogenetic part. This is very intriguing. And when I food rationed the chicks of the 2 lines, with a quite complicated and demanding protocol, I had hundreds of chicks in the lab, all fed in a standard way by 20 students and myself, of course, from early morning till the evening for months. And then we tested them, and well, you see, this is the exploration score, the selection trait. This is the parental generation, and in the next generation, we see an interesting thing that they all become fast, both control and full ration. Well, very interesting, but why the control and the full ration at both? So there is no effect of the treatment. So this was at the same time very interesting because these were genetically selected. So when you know the genetic background, you can do an experiment. You are studying gene environment interactions, and this was a within nest design. In the same nest, we put one fast and one slow. So slow birds became fast. Fast birds were still fast. There was also a long-term effect. It's not shown here because 6 months after, the line differences reappeared, but controls were also affected. Well, we did another year, there were problems that year, so an experiment in which we used a between nest design, and indeed, control and food ration chicks differed. So the food ration of chicks. So what is this? This food rationing, I mean, it's not, it's very mild. So it's having, we measured, we with accuracy, we let them, it's like if for 3 hours per day they receive less, a number of 3 hours of feeding, the feeding rate was about 3 hours per day less in this animal, which is something which can happen in nature. There are the good years and the bad years. I know very well when I go into the fields, when there is a bad year with bad weather, a few larvae of caterpillars or so, you go, and as far as you go, they start to beg immediately. They are hungry. The sibling competition is high. And this can affect, of course, later behavior. Anyway, this study, which I summarized here, showed that personality can change across generations, mainly because the environment varied. And we could show a 3-way interaction in which the genes, the genetic background interacted with the physical environment, so the simulation of a bad year, and the social environment of the brood, because when the chicks were together, there was no effect of 2 rations. You know why? We show it in a separate experiment, that because one chick, even if it was not food rationed, was influenced through the mechanism of social facilitation by the chick that was hungry. So he also begged. So one was screaming, the other also was screaming. and became fast as well. Well, what could be the functions? This is speculation. A fast strategy may be better in adverse food conditions, clearly. It may be also adaptive. I mean, if the higher exploration scores to disperse after experiences adverse food conditions. And then we show it also that, and I haven't shown it, that aggression was decoupled from exploration, because in those individuals, these individuals were not, here, the line difference is not anymore present, but the difference in aggression was still present. So probably aggression has stronger, has less flexibility, stronger genetic component. And this decoupling, what I call it ontogenetic decoupling, suggests that personalities do not pose a so necessary rigid limitation to behavioral plasticity. Other studies in parallel showed that the two types can fluctuate across years that differ for food availability. And this somehow put together this laboratory simulation with the findings by Niels Zingermans in the field. Well, what could happen? I haven't studied this, but I have to tell you that epigenetics and now epigenomics, I mean, that's the main mechanism that could be at the basis, at the roots of stable behavioral responses. Through epigenetic regulation. Just to mention this, there are many studies on, especially on laboratory rodents, that show that maternal care, parent-offspring interactions, and the early environment has a strong effect mediated by epigenetic regulation at the molecular level. But as an example of epigenetic regulation, I want to go quickly into the issue of the maternal effects. Well, we know that yearly exposure to maternal steroid hormones it's a phenomenon that occurs in oviparous species, but not only maternal steroid, also the placenta releases. So the testosterone is not something of the males, of course, only, and that this can have long-term organizing effect. They could affect the differentiation within the same sex, and this brings back to the Japanese quail, and they influence the pleiotropic effects. Well, let's focus. Well, especially birds contain substantial levels of maternal androgens, which vary, they're not randomly distributed across the clutch, the eggs, but they vary systematically with the position of the egg in the laying sequence. And this can be affected, so the mother is a kind of interface and then the levels can be affected by environmental and social factors. To what extent they is still debated. I mean, the effect, the long-term effect of maternal hormones is not completely clear. And especially also the mechanism, because then these hormones, they interact with the endogenous hormones that the embryo produces in the egg. Well, at that time, we decided to measure the maternal androgens in the eggs, and that's the result. The result was quite surprising. I mean, the line did differ, but the lines differed with a kind of interaction with the laying sequence. This laying sequence is the number of the eggs. So this great teats lay 1 egg per day. And after the last egg, they start to brood, to incubate. Well, there is an interpretation of this, but that would take too much time.